record. And it's recording. All right. Thank you. Take it away, Mark. Welcome.
Hi. Hi, everyone. I've been participating with Wendy on the membership committee for about 2 or 3 months now. And it's been a really interesting and rewarding experience. I've enjoyed working with her and also the other members of the group. We've been sorting out some feedback from the larger group of master beekeepers. And I've also been able to engage in some topics that are of particular interest to me too. So those two topics have been some of the ways that the outreach can be quantified and possibly organized in a way for comparing how effective these different outreach efforts are. But also another interest of mine has been honey analysis. And so I was able to write a blog. And Wendy, thanks so much for that invitation. And I'm happy to be doing what I'm doing.
Awesome. Thank you. Philip is also with us. Do you want to share a couple of words, Philip? Maybe I'm putting him on the spot. I'm not sure. I can unmute you. No, I cannot. But Philip is our social media expert, so I'm really looking forward to working with Philip. I got to tell you that. Yeah, there you are. Thank you.
Sorry, it took me a minute to get unmuted. I have
Yeah.
So just kind of wanted to echo, I think that the membership committee has been sort of a great opportunity for us to get together to sort of discuss issues members are facing or questions that members have. And so we would like to encourage everyone to reach out to us as well. We know that sort of Wendy and Elina and Bernardo all have a lot on their plates, so we would love to kind of step in and help where we can as well. So I've been excited to be a part of this and looking forward to what we're going to get done.
Yeah. We're happy to have you on the team, Philip. Thank you. And it's important to hear your voice. This is part of, we're running with a lot of the comments that we received at the town hall. So we're actively working on answering your questions, as you saw in the newsletter. So again, thank you for showing up tonight. And I am going to turn it over to our expert in the house, Dr. Elina Nino. Take it away.
Thank you. I feel like a rock star. You do that. So today I wanted to do something a little bit different, and I realized that we talk at you quite a bit, and I don't know how effective. Hopefully it is effective to a point, but we all know that learning is just plastic, if you will, and people learn differently in different situations. We also got some feedback before from some of the members saying that they would like to have more of an opportunity to sort of do discussion type webinars or discussion, I guess like cafe chats and something similar to that. So I thought for tonight we could try this out and see how it goes. And it really is a more active way of doing a webinar, I guess, instead of you just sitting there passively listening and potentially even falling asleep. My old advisor was joking, it's nice when we can see everybody so we know nobody is over in the corner licking the walls or doing something weird. So now is the opportunity to have a more active webinar where we can all together actually go through a couple of articles that I've sent earlier, and I'm hoping that most of you have received those. They're fairly short articles. There are 2 articles that are actually media articles from a couple of different media outlets, and also a couple of articles that are actual research articles that these media articles were based on. And one of the reasons when I get asked to talk about how do we interpret or actually spot, I guess, bad sides, it's most often the question. the main reason for the general public usually is, well, how do I know that what I'm seeing in the news is actually true? So we can go over a couple of things that have sparked a lot of controversy, I would say, among the general public, and also outcry among general public and also among researchers. And those two articles are the study links B decline to cell phones. And also the second one is Honeybee Mystery Solved. Harvard research finds direct link between colony collapse disorder and insecticides. So that's actually one of the first things that we're going to discuss tonight. And that is, let me just pop up one of these. I said no PowerPoint, but really I think this will be useful to have in front of you as we're going through it. Of course, if there we go, screen would behave properly. So let me just share this again with you before we go ahead and get started. Share the screen. There we go. And hopefully everybody can see the screen now. So I'll just, instead of going, as I said, through a PowerPoint per se, this was actually something that Wendy found online and thought, hey, this is kind of cool. It's a sort of a guide to spotting bad or really poor science. So the first thing that I already talked a little bit about are these sensationalized headlines, whether that's a media article or an actual research article itself. As soon as you hear things like final mystery solved, or it's a direct link for sure, and now we can forget about everything else, and we for sure know that cell phones are causing the die-off of honeybees, so let's all get rid of our cell phones and cellular networks. And again, this is simply because the reporters want to sell either newspapers or their blogs or online presence. So the more dramatic or sensational the title is, presumably the people will be more attracted to it. Unfortunately, when you go into those articles, oftentimes, and now of course everybody can be a journalist, oftentimes you will find not only that they sensationalize things in the article, but they quite often don't actually have the proper research background to evaluate the research behind what they're reporting on. And that often leads to articles that really misrepresent the results. It really can take everything out of context, and they'll focus on a single line within the paper, within the research article, that claims we now found, for example, that neonicotinoids are the cause of colony collapse disorder. And they'll often read an abstract or just basically the conclusion at the end, and that is enough for the reporter to now tell the world, oh, we've solved the problem. Right? So one of the things that I often will tell my students when I talk to them about this is, don't just rely on the news article, go and find the original research article. And that original research article, there is a big push for researchers and scientists to actually have open, peer-reviewed, accessible articles for the general public. So that used to be, I think that it still is an issue for quite a few journals, but one of the good things that's happening from all of this is that there are more and more journals that are offering open access articles. So you as a reader and, you know, a taxpayer, if you will, have access to that information directly from the research articles for free. Before, I guess, we go into depth of these two articles, couple of things that actually came up as I was looking through some of the I guess, rebuttals to a couple of these articles to keep in mind is that we have to think about potential conflicts of interest within these articles. And for example, the article that is talking about neonicotinoids, it apparently has become public knowledge that the gentleman or the professor who actually published this research gets a lot of funding apparently from an organic farming company. So that's one of the things that why we as a researchers will always have to write down, and we should be writing down and reporting anything that would potentially be seen as a conflict of interest. So if I'm, for example, testing anything by Bayer and Bayer is paying for it, we need to disclose that, who's paying for it, and also we need to state that there is no impact of the funding source. Hopefully there is no impact of the funding source on any of the results. Couple of things oh, sorry, I totally clicked on it. Okay, so that's 3. 4, one of the things that people often confuse is correlation versus causation. And there is a famous graph or famous graphic that was popping up everywhere throughout the hallways of all of the universities that I've been in so far. And it shows the correlation of the, I think it was the climate change increase in temperature in the world directly correlates with the decrease of pirates, I think, in the world. So clearly there are a lot of correlations that you can show in the world. However, that doesn't mean that the climate change is leading to decrease in the pirate populations or vice versa. So in order to show causation, you actually have to often follow specific rules. For example, you can select a stressor and make sure that you apply it appropriately, and then see what happens with, in this case, obviously we're talking about honeybees, but see what happens with the honeybee. Does it die? Is it that what caused it? So after you go into, let's say, do a bee-topsy, I guess, or an autopsy, you have to be able to show that, for example, a pathogen has replicated in the bee, and oftentimes the researchers will take it a step further. They will take that bee that died from a pathogen, and they will actually feed the solution from that bee to another bee to see what happens. So there are quite a bit of steps that a proper researcher will take to show that something is not just correlated, but also causes different maladies, for example, or mortality or morbidities, and has different symptomology in honeybees. Right? Okay, this is a tough one. Number 5 is a tough one, and it's talking about unsupported conclusions. And we'll often see this, and actually I will often see this in really young researchers that are just starting and are ready to take on the world and solve all of the world's problems. They will take their research results and they will often speculate inappropriately that their results mean one thing where there's really not enough support to claim whatever they're claiming. And we'll see something like that in both of the articles that we're going to be talking about. We will also, in both of these articles, see huge problems with sample size. So huge problems with sample size are very common, and especially can be very common with research with honeybees, for example, because it's really difficult to process, so to speak, a large number of colonies. Large numbers of colonies meaning hundreds of colonies that you could be using per treatment, and you'll hear more and more from beekeepers and other researchers that you only used 5 colonies. That's completely inappropriate. you need to use at least 10 times more. Of course, that's not always possible, but we definitely need to consult then at that point a statistician or have a little bit of a working knowledge of statistics. And oftentimes researchers will actually start with determining what we call doing power statistics. And that power statistic will often suggest what is the sample size that a researcher should use to appropriately make conclusions from the results that they draw. So again, problems with sample size, I would say, because normally, probably, unless you have statistical background, and if any of you have statistical background, I definitely have some stats for you to work on if you're interested. But when you were talking about statistics, again, if you don't have statistical background and you don't have a statistician in your life, sample sizes, it's safe to say that anything in terms of colonies that is below 10 colonies per treatment, especially if it's not replicated over time, is not sufficient. So that is the bare minimum that the researchers should strive towards. Okay, this is more, well, number 7, it's more often that you'll see this in human trials, that unrepresentative samples are used. So, for example, if you just stick with, I think we've heard quite a bit of this commentary recently on understanding heart issues in women and how we really don't understand very well what these heart issues are and how they're the symptoms of, for example, heart attack can be very different in women. And that reason being is that quite a few research studies in the past, and probably still, that have happened have used white males. So what a white male experiences might not be the same what a white woman experiences or any of the other minorities. So always using a representative samples would be good. In case of honeybees, you can argue that using groups of different subspecies or different stocks of honeybees or a mixture of those could be a representative sample, more so than just your average Italians. Or on the other hand, you can argue that sticking to the same genetic background will reduce variability. So oftentimes researchers will do research on a couple of different stocks concurrently. This is a big one, number 8. And this is something that it's not necessarily, I guess, innate to everybody. And this is something that's been drilled in my head ever since I started in college working towards a science degree. Control groups. You need to have a control group. For example, in humans, they have an example. Obviously, you have to have a placebo. Right? For example, if you're testing a drug, you should have also a placebo to see if it's really the drug or if it's just the state of mind of an individual. In case of honeybees, and again, we'll talk a little bit about that in relation to the 2 articles that we read. In relation to honeybees, oftentimes, for example, if we are testing, I don't know, a novel miticide or novel miticide that potentially could have use for control of varroa mites, we don't only use a no treatment control. We also need to use a control of what we call a positive control. So the control compound that we know has been proven to reduce varroa mites successfully. So it's not always quite as clear what a control is, but knowing that it's not just one control usually when we're talking about research, it can be in some cases, but knowing that it's not only just control, one control that needs to be used also is very valuable to keep in the back of your head, in the back of your mind. No blind testing, so we're down to number 9, no blind testing. I assume everybody understands what that means, but basically Oftentimes, especially if we're working in groups, what we will do is one person will set up the treatment, and then another person will take the measurements without knowing which colonies have which treatment. So that's quite important, because even if we don't think we're being biased, subconsciously we can be biased when reporting results. So we highly recommend, and we are during peer review process always asking about a possibility of doing a blind test, basically. In terms of selective reporting of data, that can also be a problem. That's a little bit tougher to spot if you just have a research article in front of you. Of course, you don't necessarily always know if there are any additional studies that the researcher has done that might not match what they're reporting on. More blatant instance of that is if they report the results and only focus on drawing conclusions for a subset of those results, especially if it doesn't match what they're sort of are after and what they're hoping to accomplish. This is also another big potential issue for scientists and researchers, and these are unreplicable results. So basically, when you write a paper and report your research, you need to report all of the materials and methods that should be able to allow somebody else, if they choose to do so, to replicate the study fully. And for example, or to replicate the study fully. So if somebody chooses to actually do this research again, or they want to repeat the research and follow up on it, and that's the reason they're following up, they need to be able to get the same results. Now, this can be a tricky thing because there are a variety of things that are happening within the environment, especially when we're talking about honeybees that are sitting in San Joaquin Valley here in California might not have the same either genetic background or the same environment as in, let's say, Pennsylvania. So you might end up getting different results if you are trying to replicate the study. However, one of the things to keep in mind is that if you read through the papers, researchers will, they're very aware of that, will often state that under these specific circumstances, ... these are the results we got. So it doesn't mean that they're not necessarily always false, but it could be that they're not reproducible in a different environment because simply the bees behave differently. They're exposed to different potential pathogens, different stressors, for example, lack of food, increased veromite loads here in California. So that is one of the things, of course, that as a researcher or a reader of the scientific paper, you might not be able to always evaluate, also because if nobody has replicated the study, then you can't tell, oh, it's not reproducible. Lastly, the papers or the research articles that are out in the public before they are published, and this is why universities and institutions value peer-reviewed research more than, I don't know, extension articles, for example, is because it has been checked and looked over by somebody else, another researcher. When you go through the peer review process, you usually submit the first draft. The editor of the journal will look at it and say, okay, this looks like it is appropriate for our journal. And then they'll usually send it to about 3 reviewers who are also experts within the field of whatever you're submitting the research on. So somebody else, before that study comes out, somebody else has looked at this research, and at least 2 out of 3 people have agreed that this is publishable. It was properly designed. The experimental design was appropriate. You had enough replication. You're not claiming causation just because you have correlation. They check your controls, they check the blind testing, they make sure that you're not overstating your results. That again is a big one. That's oftentimes, it can be frustrating because as a researcher, again, you want to make sure that people are aware of your research and you're excited about it, but you also need to be aware that you shouldn't be overstating your claims, especially if the results don't support that. So peer review is really, really important part of the scientific publication process. And it's really important, and it's also, I think, underappreciated, because we as scientists and researchers are expected to provide this professional service to our colleagues, to our peers. Oftentimes, well, I think all of the time, we don't actually get compensated for it necessarily. But it's quite crucial, and I think this is one of the things that we as a scientific community are really need to embrace and just keep doing this, because otherwise a lot of stuff, it still slips through the cracks. A few studies here and there are not found, and you'll see when we talk about the next two studies. But this is definitely something that the scientific community supports, and as a scientific member of a scientific community, we take this very, very seriously, and we'll often spend hours on a paper that sometimes just doesn't make sense. On the other hand, there are those papers that make it super easy for us to review them because everything is done properly, and we love those. But again, peer review process is quite crucial. So before you even see the paper, there have already been at least 4 or 5 independent researchers that have read it. Again, doesn't mean that, you know, things don't slip through the cracks. So you should always be aware that there still might be some mistakes potentially in there, but it's already gone through a pretty rigorous scientific peer review. So by the time the paper is published, it should be pretty well vetted. However, that's not always, as I said, the case. And some of the things that are not necessarily on this chart that you should be thinking about as you're going through your sort of critique of various research papers is, what is the research background of the scientists that have published the study, for example? What is the scientific reputation of various journals that these articles have been published in? There are so-called, what we call predatory journals, or what they call money-grubbing journals, that will publish pretty much anything without necessarily appropriate peer review process, as long as you pay the money for the article publication. And that's not the way things should be working. So again, you should always look at where this journal stands amongst other journals within the field. And that's really easily checked. You can just type in the journal name and look at sort of the, you can put in an impact factor, and you can just sort of look at it and compare what type of other research has been published in there. Also, other researchers who has published there should be a dead giveaway. For example, if you don't know any of the researchers in that publication, and you're working with honeybees and have gone to many conferences and many scientific gatherings, so to speak, and beekeeper conferences, yet none of the names that you're familiar with in terms of researchers are in those publications talking about honeybees, for example, that could potentially mean a red flag. Okay, so what we're going to do now, and before we dig deeper into some of these media articles, which I'll pop up, I wanted to see if any of you had questions or anything that wasn't necessarily clear. And I can't get out of my... Nobody? course, my computer froze. Oh, there we go. Okay. No questions? Everybody knows how to review articles? Perfect, because we'll do that. You can, so you should be able to unmute yourself.
Hi, Elina. It's clear here. I like the list. I haven't seen that.
Mark, you just had
Hey,
a bit of
Mark.
feedback. Yeah, so we just muted you again. Sorry.
Okay, Mark, I'm gonna unmute you again.
Hello.
Okay, that's better.
I can, Wendy and Elina, I can also type. I'm following the chat board, and that's a nice, easy way for me to write in questions. But I just wanted to confirm that was clear what you were describing, and I think a good introduction. You're right, there's a lot of depth in each one of those categories as you get more involved in the field. And those of us that even have published in different disciplines have their own criteria, as you described with the statistics for beekeeping, are quite unique.
Yes, absolutely. And then Wendy, can you help me? So John is right. How can we contact them? I thought there was a way for a participant to raise their hand.
That's if you have the webinar feature, and we do not.
Oh.
Yeah.
Oh, get that. Okay. So either way, I think if you have any questions for now for this specific topic, what you can do is just maybe put in a question mark into the chat, and I will unmute you. I think that will be the most effective thing because there are quite a few of us. And for now, I just wanted to get a feedback from all of you. And again, you can use the chat feature. Just type in yes or no if you have received the articles and had a chance to look at them. Oh no, not the cell phone one. The cell phone one is the most fun. So the PDF that you've received should contain both of those, all 4 of the articles, Elizabeth. So maybe look up and down the PDF. Okay, looks like most of you have gotten it. So what we're going to do now, we're going to focus on the first article, which is the easy one to critique, hopefully. And that is the one that is speaking about the effects of cell phones on honeybee colony collapse. So, for those of you, and it seems like most of you have actually had a chance to read it, and based off of what we just sort of talked about a little bit, what are you able to tell me? And if nobody volunteers, I'm going to be like a class. I'm going to start calling out people. But what were you able to ascertain from the media article and also the actual research article that was talked about? Okay, so let's hear John Johnson. I'm going to unmute you. And John, you can tell us a little bit about, you mentioned unsupported conclusion, and I'll unmute the John Winsler as well because he mentioned small sample sizes as well. So I think I unmuted John Johnson. John Winsler, unmuted. You have the floor. gentlemen. Yeah, I can't hear you. That's true.
Elina, Amy Houston also said small sample size. Maybe Amy, you can unmute yourself and speak to that.
Can you hear me?
Yes.
Okay, so the sample size of the study about I can't say it, neonics, said that they used 18 colonies. And then they further subdivided that into 2 different groups, feeding 2 different types of sugar syrup. And then I think they further subdivided that. So 18 is not very large at all.
Yes, so that's one of the 18 colonies was the neonicotinoid one. The other one, the cell phone one, which is the one I wanted to start off with, is
Oh, right.
the one that had, I think, only 3 colonies.
Yeah, so that's even smaller than 18.
That is way smaller than 18. And I think it was surprising to quite a few of us to even see that come out. And then somebody like, I think it was CNN that actually took it and published, or the story was published on CNN, I think, .com. So that was surprising. What else, when you're thinking about this study where they placed 2 cell phones, just a review for those of you who haven't had the chance to read it, placed 2 cell phones inside the colony. It was a 7-frame colony to start off with. They at least had appropriate controls. So they had 2 working cell phones. One colony had 2 non-working cell phones, and then the last colony didn't have any cell phones. And then they turned on cell phones, from what I remember, 2 times per 15 minutes, twice per week. So if I pop up the picture of the actual setup, I think it will be clear to everybody why I sort of chose this article to discuss at length with most of my classes. So let's see, share. There we go. Everybody sees that, I hope.
Yes.
And I'm also, yes, and I'm also hoping that everybody is nodding their head and shaking their head and saying, oh my God, why would they do that? So what do you see wrong with this in addition to the super small sample size? Maybe something that we haven't discussed yet.
This is Phil.
Mm-hmm.
One of the things I would say is that that's not typically how we would have a hive. Right, it's missing 3 frames, which
Uh-huh.
alone is different than what we would normally be looking at. And I'm not quite sure what the box on top there is, but it looks like maybe there's not even a bottom board in there.
Yeah.
So it looks like
And
perhaps the actual setup is not typical of what would actually be going on inside of the colony.
yeah, and then normally would you have cell phones inside the colony?
No.
No. So it is really completely, like, this is sort of the worst, I think, of the worst science, if you ask me. In addition to small sample size, they, at least as I said, have appropriate controls, but this is not what you would normally do in a colony. So as a beekeeper, you don't have your colonies outfitted with 2 cell phones, and then you put a huge empty space, and then, as you said, just 7 frames. I mean, arguably, I guess you could have a 5-frame nuke, but not a huge empty space there. What else can you see or point out that you wouldn't buy this study, or hopefully you wouldn't buy this study? What other things do you notice?
Hi, Elina, it's Mark.
Yes.
Right, the screen that you have it on, that results table, I guess I noticed that some of those treatments are there. The standard deviation is big enough that they look like they overlap. In fact, so the conclusion that they're really different is almost a wash.
Mm-hmm. Absolutely. And then, so what else in terms of the numbers and looking at the standard deviation? Did they even talk about statistics? They don't. I don't think they talk about statistics at all, actually. And that's another big red flag. If you're not necessarily talking, I don't think they were talking about it. If you're not talking about statistical analysis, which basically draws conclusions based on the sample, draws conclusion about the population based on a smaller sample, then it's really just not even, it's not useful at all. Especially if you have only 3 colonies. Can you have an average of 1 colony per treatment? No, you can't. Just so you know, you cannot have an average of a single colony, right? If you have 1 colony, there's no average. So it really is, again, small sample sizes, inappropriate experimental design, no statistical analysis presented, so we don't know what they've done. And what were their conclusions?
Elina, this is Mark again. And I'm just reading, I'm still looking, trying to find some methodology and how they do have a p-value in the abstract, and they're saying that there's a significant decline in colony strength, but I agree, I'm still trying to even find what that is in reference to. So they're calling it a decline in strength, and that's an interesting variable, I guess, in and of itself, that again might be defined in methods that I haven't quite figured out yet.
Yes, I don't think I've read this several times over the years, and it's still the greatest article to use to get people sort of get their feet in understanding the appropriate research, and what are some of the things to look for. So what other things would you say are fishy, I guess, when you were looking at this article?
I didn't recognize Current Science as a journal. I don't know if that's just my limited reading, but is that one that you'd heard of, Elina? Current Science?
Never. Never. I've
Okay.
never heard of it. I've never heard of it. Yes. So again, that's another thing to keep in mind. And then going back to the actual the media article. Now I'm going to talk about this. I know everybody's waiting for the NeoNIC study, because I'm sure you will have a lot of feedback probably on that. If you look at the media study, it just keeps on going about how study links bee decline to cell phones, parasitic mites called varroa, and the effects of climate change all implicated. But researchers in India believe cell phones could also be to blame. And then what was interesting, I thought, if you were looking at the media article, is, and that was actually really good and helpful, that the reporter, in addition to reporting on the research, has actually contacted 2 different researchers as well to get their input, which I thought was very valuable, but they had sort of the opposite response. And that also is another thing that, again, is a good thing to hear sort of both trains of thought, but neither one of them explicitly said, yes, this is for sure what's happening, but rather the other one was saying, oh, it doesn't, it could be, but it's not really something that has been definitively proven. Okay, so let's move, and please, I think I was trying to get back to the chat feature. I don't know if I can see chat feature when I'm showing my screen. But please, as I said, don't be shy. This is an opportunity for you to, as I said, exercise your brain cells and neural connections. And it's also hopefully helpful moving forward when you see, of course, there's a lot of reporting on honeybees and pollinator declines. So hopefully this will be at least somewhat helpful to, if nothing else, get you started on thinking and critiquing the research that comes along your way. So this is a study that probably a majority of you have heard of, if nothing else, have read about it in the media. And it has caused quite a bit of uproar among environmentalist groups, especially. And I see there were some of the comments on disrupted the colony, correlation versus causation issue. You're absolutely right, John, and both Johns. Overhead power lines are not the same as cell phone EMR, exactly. The bees seem to be confused. It really is supposition and anthropomorphizing. Time of the year also could have been affecting the egg laying. So you're absolutely right. So thinking about this is great. You guys are thinking about all the right things when it comes to at least the first one. Okay, the first article. Okay, so this one was a little bit tougher, as I said, going back to the NeoNIC study. And again, it might have been something that you've heard about. It didn't happen quite that long ago. I guess it's still only been 5 years. But here's the, let's see. There we go. Honeybee mystery solved. Harvard research finds direct link between neonics and colony collapse disorder. So let's just start there. Let's see. Lou published another article that was very misleading, and Randy Oliver called him on it. The article might have been retracted. It is the same guy, John Winsler. It's the same guy. It is, oh goodness, what is Chensheng Liu or Alex Liu? And he published another article prior to this one in 2012. I don't think it necessarily garnered as much attention as the second one, and I'm not sure why. It could have been just the fact that the reporters, when they were looking for something to report on, they just missed the article. But the second one, which is this one, the 2014 article, was definitely the one that kind of was thrusted into the media world. And if you're just looking at the title, what do you notice? It's the first thing on our little table. That's a good one too, Sandy. Honeybee is misspelled. Technically, I guess it's not necessarily misspelled, because I think in the UK they will spell it as a single word, but it definitely is, yeah, fun. And John, you're right. It's very sensational, right? Mystery solved. That's it. It's one single article based on looking at 18 colonies, solved the mystery of death of thousands of colonies. So, Amy, since you were started talking about it before, would you like to comment on the article? I'm going to unmute you, Amy.
Okay, can you hear me?
Yes.
Yeah, so it looks like they started with 18 colonies, and then they divided 6 of them out and put them in 2 groups, and they put them in 3 different apiaries. So it seems like there's a lot of variables. With such a small sample size, I'm not sure that having that many variables is a good idea. So that's the first thing. And then
you're totally right.
And then they also fed them 2 different things. They fed them high fructose corn syrup and sucrose. So why? Why do that? I mean, why? It's just adding too many variables, I think.
Absolutely. Adding too many variables, and you're right, there was absolutely no reasoning for why they did that. They absolutely didn't explain this at all. So which goes back to one of the points that's not necessarily on that list of the bullets, but when you think about it, has anyone looked at the background or the research background of the researcher who published? Or is everybody feverishly looking him up now? I'll spare you some time. He is a person, he's a researcher who up until then pretty much apparently has been researching and working with humans. So he's been looking at the effects of pesticides and environmental toxins. He definitely does not have, I think, the necessary background to conduct a sound research study on bees all of a sudden after working on humans for a really long time. And he did work with beekeepers at least, which is really nice, but also I don't know what the background of those folks is in terms of research. It's very different keeping bees, obviously, and conducting research, sound research on bees. So again, I don't think Randy Oliver was the only one who questioned his reputation, but many, many other folks have questioned this too. And I just want to see if anybody notices this. If you read the first sentence in the abstract that I just highlighted, can anyone tell me what's wrong with that sentence? Anyone?
Hi, Elina, it's Mark.
Yes. Yeah, exactly. There's Amy. Yep. Go ahead.
Oh, I'm reading Amy now too. I'm kind of curious what her
Yeah.
still lingers.
Oh, I was going to say it's hardly lingering. I don't think that's
Yeah.
the right adjective
I
or verb, I guess.
was kind of, it seems like they've already decided on what its cause was. The colony collapse disorder that appeared still lingers. Like, I guess I was kind of, I'm surprised by how definitive they are in what they defined it to be early.
Yes, and also one of the things that if you read some of the later articles, this colony collapse disorder was really very kind of localized, more so than we think. And it was not, now researchers will say that it was not present in many parts of the world. It was very specific to the US, and I think there were some reports of it in, I believe, Spain and Switzerland, or at least similar malady, whatever that might have been, that has happened. So this is, again, sensationalizing and blowing kind of things out of proportion in terms of at least where all of this is happening. And Kathy mentioned appeared in 2005 or 2006. That's the first report of colony collapse disorder, which was named later, but it was the beekeeper from Pennsylvania, whose name I can't remember now. I'm sure some of you remembers it, but he reported the massive loss of colonies in his apiaries, and the issue was that none of the bees were still remaining in the colony. All of the worker bees were gone, and seemingly just the brood and the queen were still there. So that was why it was a huge deal with, like, oh, the mystery of the disappearing bees. Yeah, something like CCD in the Isle of Wight just prior to 2005. That's right, David Hackenberg. So throughout the, tracheal mites. Throughout the years, there have been, I guess, a few instances in which colonies have collapsed in the past, whether it was due to, people think it was tracheal mites, or it might have been American foul brood as well. But it has happened in the back. But specifically, colony collapse disorder was that instance of when David Hackenberg reported that he lost, 50% of his colonies or something like that. So again, sensationalizing whatever the findings are. Okay, so let's move on into some of the other aspects of the article. As you read through it, what kind of struck you as odd or not odd? So, Kathy, this was their reports or their research was based on, well, that is what prompted them to do this research. The huge impact that the colony collapse disorder reporting had on really policy making, as well as the general public outcry for more research being done on this issue. So that's why they've done it. And colony collapse disorder is still being reported from some of the beekeepers that are doing the Bee-Informed Partnership Survey. But that's what prompted them to do the research that they did. Any other input? Mm-hmm. Absolutely right, Amy. They tested for mites, but they tested for nothing else. They were in 3 different environments. They were in 3 different apiaries. They had 2 different sugars. I don't remember, now that I'm thinking about it, I don't think I saw any reporting of feeding pollen or protein. And we know that pollen or protein are necessary for stronger immune response. Don't really know much about the background, I guess, of the bees either. I don't think they specifically specified of what the hives were, where they came from, the background of it. What about the neonics and anything weird there? Yes, John, you got it. There was a big critique of this article, specifically looking at the dosages. They were overexposing the bees to the neonicotinoids. Basically, they fed them continuously for 13 weeks. And unless the colony is in pollination of crops that have neonicotinoids applied continuously for, 3 months, then this is really not what you would normally see out in the field. So thinking back at the table here that we have, spotting bad science, does anything else come along, or does anything else strike you? I would say unsupported conclusions, definitely problems with sample size. People have tried to replicate the results, and I'll tell you a little bit about this in a bit after the study was published, and they were not able to. I don't know about the selective reporting of data because, again, I don't know what else might have been done. They didn't talk about blind testing at all. Somebody mentioned that another control group could have been a positive control in terms of another pesticide that we know would cause mortality. Again, samples, we don't really know, again, what the hives, the genetic background is. And they're drawing pretty large claims and relying on bad statistics, actually, to draw conclusions. I heard somebody unmute themselves, so.
Yeah, Elina, I'm here. I'm looking at that table, and I wonder about what results they chose in this response variable of the number of frames. I thought I read mortality in the mortality as being the final conclusive information in the abstract, but now I'm looking at the data in the table, and it looks like of all the variables that they measured, this number of frames, reduction in number of frames, seems like it is dramatic, and it does argue in the direction they were hoping for. And so maybe that's a promotion of a result that they were favored to and maybe selective for.
Yeah, exactly. And they do talk about mortality as well, as in 6 colonies out of 12 that were treated with one or the other neonic died. So I don't know if they mentioned which colonies died, whether it was those treated with imidacloprid or clothianidin, but they specifically kind of group both of those imidacloprids together, or both of those neonics together. Since we're on the table that you brought up, Mark and Amy mentioned that the only thing that they've tested for were varroa mites. And that's the part of the table that talks about varroa mites. If you go down under the table and you read through what they're stating here, I always sit on this and look at it, and I just don't even know what to make of it. Varroa mite counts were not significantly different before mite-away quick strips between sucrose and high fructose corn syrup in control, imidacloprid, and neonic treated hives. So any of those, there was nothing different. It was 10, 11, 11, 10, 12, 9. But then if you read the B, varroa mite counts were significantly different after mite-away quick strips treatment between sucrose, high fructose corn syrup, in control, imidacloprid, and neonicotinoid treated hives. So the way it's worded, it's really awkward, but it seems like it's talking about how the varroa mite treatment actually worked, which is good, and there's no difference between those groups, or yes, there's no treatment between those groups. So I suppose they're trying to say that the varroa mites are not what is causing issues, because all of the colonies across the board have had the same numbers of varroa mites, which, fine, but as Amy pointed out, they didn't really look at any other potential pathogens, such as nosema or viruses, other bacterial diseases. So it's really hard to draw any conclusions just based off of that. Anything else that might have kind of jumped out at you as you were going through this? Yes, so claiming they were able to replicate their previous findings, which sounds suspicious, potentially. It's coming from the same lab. So going back to replication of the study, the group in Maryland, after this came out, it was a big to-do. Group in Maryland set out to do a similar study, and they were not able to find the dramatic declines in colonies based on neonic treatment. They also used field-appropriate dosages of neonicotinoids and included the highest possible, or the highest rate that they could think of that the bees wouldn't necessarily even be exposed to, but they wanted to use that as sort of a high point. And they, again, were not able to find issues with neonics. They also, this other study tracked everything, varroa mites, viruses, other pathogens throughout time. So they actually had additional data, not just neonic treatment. So they, I guess, essentially were not able to replicate the data or replicate the study that was done here by Lou. Anything else that stands out?
Elina, a quick question. This is Mark again. So that graph that you're looking at right now, and so mortality seems like a convenient response variable, but this idea of number of frames per colony, you've seen that measured before and used in a similar fashion. And then we're also this new idea of numbers of sealed brood counts, like those two response variables, you find that them to be good representatives of something, colony health, I guess. And have you seen that?
Colony, yes, absolutely. Colony size, it's actually something that we do pretty regularly on our colonies when we're looking at any potential negative effects. We definitely will score the frames of adult bees or frames covered with adult bees, and then we will score the brood, sealed brood area. That's only used because it's really easy to evaluate, right? So you can see the sealed brood area very easily, whereas other brood, the open brood, is not quite as easy to see in the field. So that's definitely something we've used and people use pretty regularly. If you look at the graphic that is on the screen right now, it actually, one of the things that struck me is that they've really talked at length about the average sealed brood count, but they just kind of glance over the varroa mite counts too, which is not a big deal, but again, you want to be pretty immaculate about reporting and what you're putting on your graphics.
Elina, would you normally find that those two variable sealed brood counts and number of frames with bees on it are pretty related, as in one goes up, the other goes up?
Yes.
Okay. And so the fact that they're finding that the control is what they say is significantly different than the treatment for the number of frames, and then looking at the graph with the number of sealed brood, is that, do you understand those two to be telling different stories?
Yes. Yes. So the number of sealed brood is going down, and then the number of bees has kind of remained the same in the control, as you said. Although I guess one of the things to keep in mind is that as we're seeing declines in the brood, right, the sealed brood, we're going into the winter now.
Yeah, I see the difference in the x-axis on those two graphs as being somewhat misleading, that if they only show 6 months on one and what looks like a longer time scale on the other.
Green, yep.
And so maybe that control hadn't yet deviated, which makes me wonder why they didn't show the number of sealed brood data for 2013, if that's really where they saw their difference from treatment and control as it related to number of frames containing honeybees. It would have been interesting to send that data, that the graph you're looking at, why they wouldn't have extended that out 3 months.
Into the overwinter. Yeah, and they did mention in the research, but it's in the publication itself, in the article, they did discuss the fact that those treated with neonics didn't recover and never actually started egg laying again. And didn't expand the brood again. Some of the other things that struck me as definitely not sound science, and again, this is something that you might not even realize is happening, but the statistical analysis that they did on this set of data is not appropriate. And you can look at this analysis of variance. This is what ANOVA is. Analysis of variance, one-way ANOVA, is not appropriate to use if you're doing repeated measurements on the same subject, in this case, the same colony, because the colony itself is going to potentially have an effect or an interaction with these different stressors. So that variability within the colony and between colonies needs to be taken into account. And this one-way ANOVA statistic does not take that into account. So that's an additional huge critique that was put forth by other researchers after this article came out. It was just simply not an appropriate statistical analysis. And even if you look at things like they were saying that they didn't see any effect of site, so the apiary site, apparently regardless of the apiary site, the effect was there. And then they didn't see any effect of the sugar feeding, so they lumped all of the replicate samples together. That is also incorrect to do. What you're looking for is not only the effect of specific factors, such as environment, so in this case apiary, or feed, so sugar or high fructose corn syrup, but you want to see if there are any potential interactions between the environment and the main treatment, which in this case was a neonicotinoid, whether it's imidacloprid or clothianidin. So basically, do the colonies exposed to neonics act differently in different environments? Is there an interaction between the environment and the treatment? And they didn't even look at that. They just kind of lumped everything together because seemingly it was convenient. And again, definitely not appropriate. So, are there additional questions, comments? I would like to show you one more thing before we all sort of part way, I guess. Okay, so two things, I guess, before we part ways. Couple of claims that were made in the article should have also sent up some red flags. We found, this is again overstating the results. We found that chronic sublethal neonic exposure do not appear to compromise honeybee immune system or honeybee's immune resistance to pathogens. Which, how did they know that? Did they do anything to show this in this study? Anybody feel free to say? Was there anything in this research showing that the immune response was not compromised and that they found that it wasn't compromised in this work? I'm asking you, go ahead.
This is Mark. I'm not even sure how you've spoken about immunity and different, I guess colleague in Louisiana has done some specific measurement of that. And so how do you, I don't know exactly how, I forget how you even measure immunity, immune resistance.
Yeah.
A completely different study, I suppose, a completely different study design.
Completely. And they've done absolutely nothing to support this claim. So again, this goes back to overstating the results and making false claims not based on what they actually found. And this is a pretty big red flag.
Was it biochemical that people had been measuring immune resistance before? Like you might even do it at a, not, I mean, certainly at an individual stage, if not at even smaller, like it could just be that, so mortality of an individual bee, like, is that one of the variables that you'd mentioned earlier about immune resistance?
So the immune resistance basically immunity is, you can do it by looking at various genes and expression of genes that regulate immunity. So antimicrobial peptides, you can look at for example, exposure of these bees to different pathogens and seeing what happens. They've done none of that. They didn't even check to see if there are any pathogens present in the colony. So they really have absolutely no results supporting what they're stating here, which is pretty upsetting. Okay.
Which have you and some others found the opposite of that sublethal exposure can compromise immune resistance?
I definitely, yes, there definitely have been studies showing that sublethal effects can exacerbate infections to, I think people were mostly looking at nosema, and I think they were looking at viruses as well. But nosema is easy, obviously, to reproduce and feed to the bees in controlled environments. And yes, bees exposed to certain pesticides, or at least the ones that were checked, can have indeed reduced immune response, or they're more susceptible to pathogen infections. And I can actually, if you guys are interested, I can send you some of that literature. So did anybody, what else? I'm trying to kind of get you guys engaged. I know it's kind of a different platform, but we're all in different places. So this would be a really nice way if everybody can get a little more comfortable with speaking online, even though we're not, chatting, sitting on a couch around some coffee or wine or whatever. But we're hoping to use, again, this platform for folks if they want to have discussions, if they want to have conversations, but everybody seems to be shy, maybe. And I'm not quite sure what to think about that. So hopefully, those of you who are still here, do you have any additional comments?
This is Todd.
Hi.
Hey, before wrapping up, can you spend a few minutes on kind of what is the sort of back up from the highly technical for a minute? When a researcher publishes a report or an article, like, is there a normal connection between that and sort of the larger media, right? Like, do researchers then go and try to attract media, and how do they do that, or do they do that? Like, what's the normal pattern there? And then, so that's the first part. Okay.
Go ahead.
And then I was going to say the second part is can you help us with just understanding sort of the volume of published research in any year on beekeeping that is kind of worthwhile for review? Like, it's just some sort of sense of kind of, volume, in terms of like reports and, how much is really
you would be surprised. So I guess let's go backwards. I will answer your second question first. In terms of volume of publications that come out specifically for honeybee research. I have, and you can actually do this as well, Google Scholar. If you go to Google Scholar, you can subscribe to emails that Google Scholar sends you when any new articles are published on honeybees, and oftentimes it gives you some junk too, but honeybees, bees, native bees. So I get that every day, and every day there are at least 2 to 3 articles that are related to honeybees. Every day. Did I mention it was every day? So I do not know, and I don't have a really good way of parsing out, and I don't have the time, parsing out what these articles, if they're, valid or not, and I certainly don't have time to read all of them. But again, at least 2 to 3 articles that I receive every day are based on honeybee research, and then usually there's an additional article or 2 on other bee research as well. So it's a lot. It's a huge, huge volume of articles that are published pretty much daily.
How do you decide which articles are worth spending time reading?
Yeah, so that's tough. I would start with, what are your interests? First of all, what do you want to learn about, right? That would be sort of the first step. Then I would probably, if you, for example, sign up for this Google Scholar and you get the articles, you can usually click on it and read through the abstract. So read through the abstract and see what the article is all about. And if you're interested more, then go into, I usually start with reading the materials and methods, results, and then sort of the conclusions at the end. Introduction, if you are not very familiar with a specific topic, but most of the articles that come out normally will talk about honeybee losses, and these are the factors that are affecting them. And so usually the beginning, like the introduction, can be a little repetitive. The first, and again, as I said, what's worth reading really will be guided by your interests. For example, I'm looking actually through my email right now, looking at the honeybee new results. There's an article that just came out, for example, evaluating effects of a critical micronutrient on honeybee physiology. Presumably, you might be interested in that because you'd like to improve the health of your bees. So you would click on it, and then you would read through the abstract and say, okay, oh, they're using this specific phytochemical at different concentrations, right? And towards the end, usually they will state something about the applicability of their results at this point. So, for example, this one says, the insights gleaned from this study could also advance the understanding of sterile metabolism regulation in other bee species and assist in formulation of a more complete artificial diet for honeybees. So, again, your interest, do you want to know more about this phytosterol? You can go ahead and keep reading. However, it doesn't look like that in the case of this specific article, they have any applicable results yet. So if you're just thinking about the application and whether you can use this in your colonies, then you probably will forego the article at this time and maybe wait for something more concrete. Another thing is that, for example, this article came out in the Annals of the Entomological Society of America. Entomological Society of America is a reputable entomology society. So again, this will take a little bit of practice for you who might be new to this, but in my mind, when I saw that, that's a reputable journal. So that's another reason to read the article. Okay. And another one, I guess one of the things to keep in mind is, for example, PLOS One is a big journal repository or article repository, and it's open source, so it's open to everybody for free. So you can get quite a bit of information there. And then your first question was, I wrote it down and now I lost it.
Attracting media attention. And I was
attracting, thank you, attracting media attention, yeah. So the media attention, it really depends. Oftentimes I am contacted by various media outlets to comment on articles that have just been published. Oftentimes what happens also is that you as a researcher can contact, if you're doing this at the university, for example, UC Davis. If I am interested in publicizing my research and the upcoming publication of an article that I'm particularly excited about and want everybody to learn about it, I can contact our media or the communication specialist. They will then put together an interview usually, put together an article about your research and interview you. And they will, whoever is writing the reporter usually contacts additional sources, research sources. They will hold that. If they know that the article is not out yet, they will hold back their media article and it will be published at the same time as when the research article comes out. So that's one way that the scientists can do this through the media. And again, another way is by science reporters or science writers to basically browse the newest research and find articles that might be of interest to them or research articles that might be of interest to them. And then they'll write a follow-up media article. They will contact other researchers usually, and they will put it out. But quite a bit has to do with the university also wanting to publicize the good research that their faculty are doing. So I'll often get actually contacts from the communications office asking what we're doing and if there's anything that they could be writing a publication on. Does that, hopefully that answers.
Yes, it does. Thank you.
Questions, yes. So Amy commented, I think you see it on my screen, not everyone is comfortable with statistical analysis, and I completely agree. I don't think I'm still comfortable with statistical analysis, but I think just thinking back at those, so thinking back at some of those points that we already had, you don't necessarily need to know the statistical analysis, but if you, as I said, are savvy with it, you might notice that. Let's see, what was it? Problems with sample size are usually a dead giveaway. And if a person is taking measurements on the same colony over and over again, and nowhere in their statistical analysis it says repeated measures, it usually is not good. Okay, let's see. Sandy. So that could, what about company-sponsored research to prove their point of view? Well, that is also regulated. And to a point, we need to trust the researchers that they're going to do the right thing and not do research for the money. The companies will sponsor research. If you are sponsoring research at a university, there is an understanding that, an expectation that a researcher will do proper science. And there are all sorts of non-disclosure agreements, material and data transfer agreements. University is a public institution, and within those contracts with companies, they cannot embargo research results. Their university will go back, if they have the non-disclosure agreement or the MTA agreement with a company, they will, if the company doesn't want to publish the results, the university is going to go back and say, no, we cannot say we will not publish the results. We're a public institution. We're not here to hold back results of research. However, what we can do is the researcher, once they write up the results of the publication or a publication, they can share it with you up to 30, usually it's 30 days before it is submitted to publication, to a journal. Now, that all being said, I can guarantee that there aren't researchers that might not do the right thing. And I don't know, fudge data, which is not great. But oftentimes, and I know that this has been in the news, it's not just the companies, right? It's the egos as well that could lead to submission of false data, falsifying data. So it happens, unfortunately, just like bad things happen with any vocation. But in general, again, companies that are sponsoring research, while they might want the research to prove their point of view, if they're working with a legitimate institution and legitimate scientists, that is not what's going to happen.
Elena, can you speak to the other side of that, where the university is taking a patent interest in the research, and whether you see that as creating sort of ethical dilemmas or conflict of interest?
The patents, yes. I don't have that much, well, yeah, I don't have that much exposure and that much experience with patents and the university process with patents, but when we do start our jobs, yes, anything we sign a contract, that anything or paper that says anything that we come up with, an innovation, really, if we come up with it on the university property, using university equipment, using anything that the university has supplied, that does belong to the university. And I think a person who would hold the patent would get a portion of some profits, right? But one of the big things is that the university does, within the offices of research, have a group or has a group that will work with small companies to produce the product or do a startup, for example, with a product. Now, the big, yeah, I don't know how I feel about that. I see your point. And for example, USDA, if they have any patent or if they hold any patent, they just, you know, give it back to the people. So it can be used by the people who pay taxes and supported the research. With the university, I think the idea is that, I mean, it's going back to the money, right? So if something was discovered or found based or on university grounds and with the property of the university, then they own the patent. I mean, which I guess rightfully so. The idea and the assumption is that if I just try to do this in my garage, I would not have the resources that the university offers to be able to develop whatever I might have developed. So it is what it is. Any other questions? I see a chat. I can't get to it for whatever reason. Oh, there we go. I have to put my kids to bed. Bye, Amy. My kids are already in bed, thankfully. If there's nobody, if you have a question, if you can just type it into the chat, or at least say, hey, I have a question, and we can unmute you. That would be great. No questions? So what I would recommend, and again, this is something that thank you, Ginger. This is something that researchers start learning right from the beginning of when they know that they want to be a researcher. So it definitely, I remember some of the first papers that I read, I was completely lost. Bye, John. But some of I got more comfortable with it the more I read. So what I would recommend is, as I said, this is probably quite a lot if you have not been thinking much about some of the research articles, but these are easily digested and the flaws are very obvious. So I would recommend you go through those articles, take a look, think about it, and sort of start maybe slowly reading about honeybees, topics that you might be comfortable with, and just, read through the article and see what comes out at you. Happy holidays, Susan. Thank you. So start reading articles that are not quite as dense. Highly recommend reading through the materials and methods, maybe even results, then materials and methods, and then the discussion. And think about some of those telltale signs of potentially bad science. The last thing, for those of you who are still remaining here and hanging in there with me, I wanted to show you, and I don't know if I'm sharing the screen anymore, I don't think I am, but I wanted to show you one last thing about this neonic article. Somebody was mentioning that Randy Oliver was very critical of it. Well, if you can imagine May Berenbaum, she was livid, or at least it looks like, or it seems like she was livid after reading the study. And I'll just read this. Scientists now say that the Lou study published in an obscure pay-for-play journal proved only that feeding bees poisonous levels of an insecticide can and will kill them. So overdose. University of Illinois Chair May Berenbaum has called Lou's research effectively worthless to serious researchers. The experimental design and statistical analysis are just not reliable. He never tested for the presence of other pathogens, so his conclusions dismissing other likely causes don't follow from this data. The whole study just doesn't hold together. And she points out one thing, which again was quite disappointing, is that it came from Harvard. So it does give it the credibility in this case that it doesn't deserve, which is again very, very sad, and it really did garner a lot of attention for people who didn't necessarily understand that the science really was just flawed behind it. Okay, I ended up still talking most of the time. I apologize for that, and hopefully, as I said, we will find a way to get used to this platform, and we'll see if we can get that webinar addition to it so we can have some sort of, I know the order is difficult, but we can maybe get the webinar platform or webinar edition so people can raise hands and we can unmute them as they have things to say. But again, this is something that you will get better at the more time you spend reading through the articles and even just looking at or listening to various presentations from various researchers at beekeeping conventions or beekeeping meetings. And if you have any additional questions, again, as I said, this is just to get your feet wet and thinking about research and how complex and complicated it can get, and don't just take it at face value. As you think of the questions, feel free to email me, and I will do my best to answer. And also, if you are interested in any articles that you would like to read and you can't get them through open source journals, just let me know and I can get those to you. Okay, I see one chat. I'm guessing people are probably saying good night. Yes, thank you.
Yeah, thanks. And
thank you, Mark.
And if I could provide just a little feedback on the forum too, I think we're all kind of charging into this territory of talking to our computer rather than each other. I think you're making an honest effort, and I'm optimistic that conversations and discussions will develop as time goes on. Maybe the subject matter and other things will be good catalysts for future discussions.
Absolutely, and again, that's the hope, and I totally understand when I first started using this platform, it was very weird, but with time, again, we kind of got used to it. Thank you, Todd. As you said, I'm actually talking into the computer and into the phone, and it can get awkward, but I would like to just encourage everybody to go for it, because I think this is going to be crucial for having discussions where we don't all have to be in the same place.
I think so too. I think it's a powerful tool. Cheers. Have a good evening and happy holidays.
Happy holidays, everyone. And again, thank you very, very much. And questions, just send me questions and hopefully I'll be able to answer them.
Take care. Good night.
Good night.
Good night, everybody. Thank you for attending. I'll send out the recording tomorrow. Thank you, Elina.
Okay. Thanks, Wendy. I forgot we were being recorded.
Take care.
Perfect. Bye.
Bye.